Kia ora koutou. Like John said, I'm Jo Carpenter if you've just arrived. And in this talk, I'm going to be talking about our research, which examines shifts in shipwrack diet during and after a large beach seeding event in Fiordland. Before I kick off, I'd just like to really thank all the people involved in fieldwork because most of it involved bagging up dead rats. So it wasn't very pleasant, but we really appreciate your effort. Thank you. So this infographic here was created by Doc, and it's often used by their comms team, especially when pest control operations are happening in response to beach masts. Obviously, Susan's just given a lot of nuance around how pest mammals respond to beach masts, but I'd say this kind of sums up a very high level understanding of the processes going on in these years. So in the beginning, we get lots of beach seed in the late summer or autumn. This results in lots of rodents. Then we get lots of stoats. And in these two latter stages of this infographic, we get the outcome that we really don't want, which is obviously those rodents and stoats then eating our native bird species that we would like to protect. But what I'd really like to draw your attention to in particular is this part of the plot circled in red, which kind of says that when the beach seed rots or germinates, we get these starving rats that have massively overshot the system, and then they prey switch to native birds, to birds' eggs and nestlings. There's an intuitive logic to this idea, but no one's actually ever tested whether this does occur. There have been two studies carried out on shipwrack diet in beach forests, but they were both carried out pretty much within the beach mast year when seed was still available for the most part. And so they didn't quite go for long enough to test whether we saw this prey switching result. So we looked at this question as part of our larger study looking at shipwrack population dynamics at Lake Alabaster in Fiordland, which Susan obviously just mentioned. This is the study site shown in this photo. The lake is out of sight, but you can see it's a classic glacial valley. You get these really strong elevational gradients. There's mixed beech podocarp forest on the valley floor. And then that grades to pure silver beech at about 500 metres above sea level, with a tree line at about 1,000 metres above sea level, because we're quite far south at this point. So in autumn of 2019, the beech trees at this site went through an enormous seeding event, which happened across a lot of the South Island, and we started following the fates of the rats there shortly afterwards. So in terms of understanding rat diet, there were several questions we were looking to answer. The first was whether the proportion of rats consuming birds changed across the mast year, the post-mast year, and an intermast year, which we kind of took to mean a sort of regular year. Secondly, we wanted to know whether the proportion of rats consuming beech seeds changed across this mast year, post-mast year, and intermast year. Then we wanted to take a more holistic approach, not kind of zone in on any particular prey item, but look whether that overall rat diet composition changed across these 3 years. And finally, because like I mentioned, we have these really strong, beautiful elevational gradients at the site, we also wanted to know whether rat diet composition changed at the different elevational bands, high versus low or mid elevation. So to answer these questions, we snap trapped rats every 3 months approximately for around 3 years at the study site, which is shown in the map over there. These black lines represent the snap trap lines, and you can see that they're kind of stratified into low, mid, or high elevation. On each of these rats, Max Harvey removed the stomach, and then we carried out DNA metabarcoding on those stomach contents. We're using bird and plant primers. So what that gives us is presence and absence data for each stomach sample of various birds or plant species. And then we went on to compare these results in the mast year, the post-mast year, and the intermast year. So this graph kind of sums up our hypotheses going into the study, and these hypotheses were born of that infographic I showed you earlier. They kind of represented the paradigm of our thinking, I guess, about the system and how it worked. So in this graph, we have shipwrack density on the y-axis. So this is using actual capture mark recapture data from the site. So these are real numbers from Lake Alabaster. We've just averaged them across the entire elevational gradient here. This is the mast year. We arrived a little bit late and the rats had already erupted, so we were already seeing them at quite high levels when we arrived. So in the mast year, we hypothesized that there wouldn't be much consumption of birds at all because rats would be focusing on the beach seeds and increasing. However, this is our post-mast year where, like Susan mentioned too, our rats have overshot the carrying capacity of the site, the beach seed runs out, and they begin to rapidly crash and starve. It was within this year that we thought we'd see a real increase in the proportion of rats that were consuming birds as they were desperate. And finally, in this last stage, this is the intermast year. At this stage, rats had gone back to their usual very low density at the site, less than 1 per hectare. And so we hypothesized that in a regular year, we thought rats wouldn't be eating many birds. They'd just mainly be eating other fruits and seeds and invertebrates. One of the reasons that this is kind of an important thing to look at is it has real implications for control strategies. So if the impact of rats just after beach seeding is mainly driven by a prey switching response because they're starving and have overshot the system, that could imply that a pest control strategy that aimed to just control them before they reach these starvation periods could have a really great effect. So you could have this kind of one quick fix almost. However, if there's not a prey switching result, and we found that rats eat birds pretty much all the time, then that would more lend itself to a maximal control strategy. Like you always need to keep your rat numbers low because no matter what the numbers are, they'll be eating birds. So here is Alex with some stomach contents from rat 479, which obviously came from winter in 2020. And we ended up with 217 of these stomachs analysed. We caught a lot more rats, but we just had to analyse a subset of them. So here's the beginning of the results. If we just take a bit of an overall picture first, what we found was that rats at the study site consumed 15 different bird species. We found birds within 19% of the rat stomachs across all elevations and all years. And in this graph, we've got frequency of occurrence of the various bird species DNA on the y-axis. The different bird species are on the x-axis, and then this has just been divided into 3 panels based on the different elevational bands. So you can see that, for example, it looks like there's a much higher diversity of bird species that were consumed at low elevation and mid elevation compared to the high elevation forests, which makes sense because that would relate to the community found at those different elevational bands. The most frequently consumed bird taxa were silver eyes and blackbirds, which also makes sense. We didn't measure bird abundance at the site, but I would imagine silver eyes and blackbirds were reasonably common. So if we get into our first question, and whether the proportion of rats eating birds changed across the 3 years. So here it's a pretty simple graph. It's just the percentage of rats that had bird DNA in their stomach, and we've got the 3 years. You can see it looks like there's actually a very steady increase across the 3 years of the study, but that isn't statistically significant. So basically what we found was no matter what the year was, whether it was a beach mast year or the starvation year or a regular year, rats were eating a similar proportion of birds. So this was quite a surprising result. If we move on to the plant results, we found across the entire study site and years of the study that rats were eating 38 different plant taxonomic groups. Again, in this graph, we've got the frequency of occurrence on the y-axis, all the different plant species on the x-axis, and the 3 elevational bands. You can see again that there's really a greater diversity of plant species probably eaten at low and mid elevation compared to high elevation. And what we found was that silver beech, gristolinia, and supplejack were the most frequently consumed plant species, which again wasn't entirely surprising. So did the proportion of rats eating beech change across the 3 years? Again, we've got a similar graph, percentage of rats with beach DNA in their stomachs and the 3 years. This is a totally different result from the birds. What we found was that yes, the proportion of rats eating beech significantly declines across the 3 years of the study. However, we still found a surprisingly high proportion of rats that were eating beech in the post-mast year and even the inter-mast year, considering that we know there wasn't any beech seed available in those years. What potentially could have been happening was that rats were eating beech seedlings through this time, which has been found by other studies by Deb Wilson. So just to quickly look at those plots beside each other, consumption of birds on the top and consumption of beech down the bottom. What about overall diet composition? Did that shift across the 3 years? In this graph, each little point on the graph represents one rat's stomach, and the different items, diet items that we find within it, kind of place it within that two-dimensional space. So little symbols that are closer together mean that those stomachs had much more similar items within them. And then we just draw a polygon around all the rats from any one year. So you can see this purpley one is 2021. We've got 2020 in the middle, and 2019, there's this pink one. And what we found was that diet composition was significantly different in 2019, in the mast year, compared to 2020 or 2021, which makes sense because we know it's a massive resource pulse in a mast year, and of course, we do expect things to change a lot for the rats then. We also did a similar analysis, but this time just looking at elevation, so whether low, mid, or high elevation rats had a different diet composition. These are the same kind of graphs in which each little point on the graph represents an individual rat, but the difference here, because there's two graphs, is mind-bogglingly, we're looking at this in three-dimensional space. So we have to show two different graphs so that we can take a different lens on that three-dimensional space that it's occupying. But regardless, we can see the same thing in both of these two plots, which is that the high elevation rats, this purple polygon, that's a much smaller space than the other two polygons. And that's what we found. That was significantly smaller dietary niche of ship rats at high elevation compared to low or mid elevation, which was an intuitive response given that it's a simpler system up high. So in terms of take-home messages, the first one I'd like to say is that rats eat birds more than we thought. We found almost 20% of rats overall had evidence of bird DNA in their stomachs, and this was higher than we expected. You might be thinking, we know rats eat birds, why is that surprising? But it is a very surprising result in light of the other rat diet studies that have been done in Aotearoa. So there's been, like I mentioned, 2 other studies done on ship rats in a beech mast year. They found between 4 and 8% of stomachs had evidence of bird consumption. And then in other forests, not beech forests, but warmer podocarp forests, there was a maximum of 5% of rats that had evidence of bird consumption. So 19% is quite high, and it could be a couple of things going on. The first is that we are the first study to have used a DNA approach to look at diet, and we know from other studies that these molecular approaches are much more sensitive at detecting bird consumption. Rats finally masticate their food, and it can be really hard to tell if they have eaten a bird. So the DNA approach gives you a little bit more insight. The other thing that could be going on, at least in reference to the studies that were done in non-beech forests, is potentially rats just do eat birds more in beech forests just because they're simpler, less resource-rich systems overall. And maybe if you're in a warm, fruity podocarp forest, potentially rats don't eat birds quite as much. Another thing that was surprising was that the timing of bird consumption didn't quite align with what we expected. We thought that most of the bird consumption would be happening in the spring when birds are nesting and vulnerable. But we actually recorded the highest proportion of rats consuming birds in autumn and winter. So there's a few potential explanations for this result. The first could be that perhaps rats are actually preying on birds at roost sites. Um, this could be particularly relevant for smaller bird species, such as our grey warblers, our silver eyes, our bellbirds, but isn't very likely for the larger bird species such as kerridu or kaka. Another potential is that we get these naive juvenile birds bumbling around and they're just not clued up yet on how to avoid rats. So maybe we are seeing more predation of juveniles as well. And finally, of course, a pitfall of any of these kind of diet studies, it could just be that rats are also scavenging on birds that are already dead. Once DNA or obvious morphological contents end up in a rat stomach, we don't really know how they got there and whether it was a direct predation event or something completely different. So there's always that to consider as well. But really, I guess the most important thing is that the study shows that it's the sheer number of rats that we end up with in a beach mast year that's predominantly driving these impacts on our native birds. So we know that rats in a beach mast year can increase to some of the highest densities ever found on the New Zealand mainland. So the impact of these increased numbers of rats is obviously going to be substantial. We don't need a prey switching response on top of that. Even if just 19% of those rats is always eating birds, that's going to have a pretty significant effect. And what this suggests, unfortunately, is that maximal control is therefore important. There's not going to be a quick fix where you can just try and suppress the rat population before we get that starvation or prey switch response. It actually just means that keeping rats suppressed for as long as possible to low levels will be the most important way to manage them. Thank you very much.
Thanks, thanks, Jo. Fascinating results. I noticed you didn't have any invertebrates in the diet analysis. Why didn't you look at inverts in the stomachs as well?
Yeah, that's a great question, John. We would have loved to have looked at invertebrates, and potentially we still could in future. I guess the beauty of DNA analyses is that we have the DNA now from all of these 217 rat stomachs, and we can reprocess them with primers for invertebrates instead. But at the time, we were just more focused on those specific questions about the bird consumption in the beach, and so we didn't open it up to invertebrates. I think one of the other issues with invertebrates is that sometimes the meta barcoding libraries aren't as extensive as you'd like them to be. So the meta barcoding will know there's an invertebrate, but they can't identify it down to a very good taxonomic resolution because people just haven't yet recorded those specifics for inverts.
Yeah, and perhaps a question that some people might be wondering, do you happen to know how long an eDNA signature would last in a stomach? You know, that if a rat ate a bird on Monday, I mean, how long would it still be there on Thursday? This kind of question.
It's a great question, and the closest thing I think that's been done is a study looking at Archie's and Hochstetter's frog DNA in shipwreck stomachs. It was a similar study, but it was just targeted towards just understanding that native frog predation. And that study, I think, calculated that within 24 hours, they thought the DNA of those frogs would no longer be detectable. So it's really a very short window if we go off what that research found.
It's a question here. Hi, Jo. Regarding the results showing higher bird eating, was the sampling method the same as the other studies, or could there be a sampling bias with rats that enter corflute tunnels being the kind of rats that would raid nests?
That's a good question. I guess Yeah, I guess ours use snap traps in corflute tunnels, whereas, for example, I think both of the other studies in beech forests, they were just using opportunistically trapped rats from DOC 150s. But I wouldn't really think that a rat that you trapped using a DOC 150 would be particularly different to a rat that you snap trap in a corflute tunnel. But it's always a great thing to keep in mind, isn't it, that we are always sampling the population in a certain way, and to consider whether that's a representative sample of the population.
Yep, Thank you. And just a comment from Dave Kelly. Nice work, interesting conclusion. The number of rats is the problem for a different species. That's stoats, that is. This was also Kim King's conclusion from her 83 paper in Craigieburn and Eglinton. So